Last October, when Dr. Victor Ambros and Dr. Gary Ruvkun won the 2024 Nobel Prize in Physiology or Medicine for their discovery of microRNA, March of Dimes scientists past and present around the world celebrated the win of two of their own. Dr. Ambros and Dr. Ruvkun jointly received the March of Dimes Prize in 2016 for their discovery of microRNA. They represent the 22nd and 23rd March of Dimes alumni scientists to win the Nobel Prize. In addition, Dr. Ambros was a recipient of the 1985 March of Dimes Basil O'Connor Starter Scholar Research Award. Dr. Ambros and Ruvkun's two 1993 papers on the discovery of tiny RNA molecules that affect the way genes are expressed, usually silencing them, began a transformative period for genetics and molecular biology. Those findings and Dr. Ruvkun's later findings about microRNA in humans opened a floodgate of opportunity for scientific research into the biology of disease and diagnostics and therapeutics for a range of conditions, from birth defects and preterm birth to cancer, heart disease, Alzheimer's and Parkinson's disease, and more. Their work has been fundamental, and I am thrilled to have both Dr. Ambros and Ruvkun here with me today to talk about their discovery of microRNA and more. The views expressed on this podcast may not represent those of March of Dimes or its staff. Information presented on this podcast is not medical advice and should not be construed as such. When in doubt, seek competent medical advice. Now, on with the show. Dr. Victor Ambros is a molecular medicine professor at UMass Chan Medical School, and Dr. Gary Ruvkun is a genetics professor at Harvard Medical School and a researcher at Massachusetts General Hospital. Both scientists join me today from the Boston area, which is also where I am based. Victor, Gary, welcome to the show.
Thank you. Glad to be here.
It's really an honor to have you. Congratulations on your Nobel win.
Thank you.
We're recording this in January 2025, so you've had a few months to let your prize sink in. Are you still pinching yourselves, or have you gotten used to the term Nobel laureate?
No.
No, you haven't gotten used to it?
No.
Yeah.
No. It's maybe.
Yeah. It's very, I mean, I know it as a fact, but it does, when you stop and reflect on it, I feel like, sort of like I'm, from time to time, I feel like I'm living a, like a script of some sort that I might have written for myself years ago, forgot about.
About to wake up in a puddle of vomit.
Yeah.
Now, obviously, there are other things going on in your lives, right? There's your families and there's other stuff happening in your labs, etc. But does this represent a very happy time in your life because of the win?
Oh, yeah.
Very much so. Yeah. I mean, what surprised me a lot is how happy this award makes the people around us. Like family, extended family, friends of extended family, get all excited. And on campus, everybody was just really, really animated and incredibly excited by this. And so then I realized that when something like a Nobel happens, a lot of people around you take ownership of it, rightfully so, take pride in it. And so much excitement, that surrounding us. And Gary, like in Sweden, everybody we ran into was so excited to see us and everything. And you realize it's not because they want to see us, it's because they're just so excited about the celebration of science that this represents.
Yeah, Sweden really bonds with the thing. They care about it as a country, and they do a good job of portraying who we are on normal TV in Sweden. And so everybody watches it, and they would stop us on the street, where's your wife? Where's your daughter? It's very, very sweet. Very sweet.
Yeah.
And then you come back to America.
Yeah.
Well, there's a lot of people who know science in Boston. I take the Green Line to work, and I would have people coming up to me and doing selfies on the subway, right? Ever since October 7th.
Yeah.
Lots of them, like dozens.
That's cool.
That
Yeah.
is cool. Victor, that might be a reason for you to start taking public transportation.
Yeah, right. Yeah. No, I mean it happens. Yeah, it happened on the airplane. Oh, it happened in Costco. Yeah,
That's so good.
yeah. So it's yeah. You asked whether it's a happy time. It's definitely a happy time, and but the happiness is I'm trying to say is not so much that that Gary and I are happy, but that it makes so many people around us, hundreds and hundreds of people, and all across the state, even are excited about it, and that's really really special and unexpected, actually.
Yeah, speaking of unexpected, so Gary, you received the call from the Nobel Committee at 5:30 a.m. the day of the win. Victor, the committee
No,
couldn't no?
no,
Oh,
3:58 a.m.
3:58.
Wow.
Okay.
Now you couldn't possibly know that, but I get a record of calls, and I said, "What time was that?" 2 weeks later, it was like 3:58.
Yeah.
Geez, no wonder I was so sleepy.
Oh my gosh. Wow. Okay, 3:58 for you and Victor. The committee couldn't reach you at first, probably because it was 3:59 when they called you, and you actually heard from your son that they
Yeah.
were trying to reach you and you had won. And I really do ask this was all with all honesty. Was there any indication, in the weeks or days before? Did you have some other people calling you or asking interviews, or was there any indication that, hey guys, you're
Yeah.
on the
No,
short list, you're on the
it,
short list, something like this?
no indication, and
No.
time to time people make comments, right? But I don't know about you, Gary, but nobody has said anything to me for probably 8 years, right? Like, oh, someday you're going to get a Nobel Prize. But what happened was I was in a meeting in Ottawa, and at somebody's students, just a week before, a student's poster, he says, "Oh, can I have a selfie with you? Because I heard you're going to win the Nobel Prize someday, right?" And I said, "Oh, that's so kind of you, blah, blah, blah." And he said, "Oh, I saw it online." So I have to admit that night I went online and I looked up, like prognostications. I thought, is this kid crazy? And of course, there was a list of all sorts of other people. And then down at the bottom, it said, "And from time to time, the names Ambros and Ruvkun, quote, bubble up." Right. And so I had to laugh, and I thought, oh, we bubble up. Where are we bubbling up from? Like the mud or someplace, or the oil spill? And so I thought, oh man. So that was, I just had to laugh, and I forgot about it. You know.
And Gary, you've said that your friends have pranked you in the past, that saying that you've won
Yeah,
the Nobel.
that was because my lab has a bit of a history of pranking, and
Mm.
so there was a time in, I don't know, 2002, I think it was. So that's 20 something years ago. We were working on a project to discover genes involved in RNA interference, which is related to microRNAs. And we were worried about competition from a very fine scientist named Ron Plasterk, who's one of the great people in molecular biology from Netherlands. And so I come to work. We've been working on this for like 3 months, and John Kim, my postdoc, slaps a paper down in front of me, and it's a cell paper, and it's everything we've been working on from the Plasterk lab, and they've scooped us on everything.
A scoop.
And
So painful.
and then as I'm reading the author list, I see one of the authors is a postdoc in my lab who's Dutch. And it's like, what the fuck? And I shouldn't swear. What the hell? And so then they look to your left, and I look to my left, and they have a video camera recording what my response is. And so somewhere there's a videotape of this. And so they had faked a paper from our competitor. And I said, you guys, you could have killed me with that. I might have had a heart attack from. So, but the labs, these people spent a week putting this paper together to mess with me.
Wow.
And so I said, if they're willing to do that, they're willing to do fake Swedish accents. And so I had been moaning about this, that these are the people who've worked with me. And so I did get a call with a fake Swedish accent. I'm not going to say who, because I don't want the person to get in trouble. But it took me in for about 3 seconds, but it was a decade earlier.
Okay. All right. Well, so no indication at all. And I'm very happy that you guys won. This discovery, like much of science, was a happy coincidence a little bit. It started with the worm, and this is a roundworm that is only 1 millimeter in length. I had to Google how long that was, and it's about the length of a sharp, if correct me if I'm wrong, but Google said that it's about the length of a sharp pencil point or a sewing needle. So we're like talking like this big. Like this is not the worm that you find in the I mean, it is a soil dwelling worm, I learned, but it's not the worm that you find in the soil that's, this long or on the street. Gary, you've called this worm badass many, many times. During your Nobel lecture last month, you said, and I quote, "We should respect their badassedness," end quote.
Yes.
Why are you such a fan? And can you find this worm here in Massachusetts?
Oh yeah, sure. These guys are everywhere, and the reason I call them badass is because the worms use small RNAs like microRNAs and siRNAs very, very well. They're kind of idiot savants about these things. And the other critters that are really good at this are things like spiders and ticks and clams and coral. And so you think of a little 1 millimeter long C. elegans, and you don't think of them as tough. But if you sort of think of it like a scorpion, if it's in the same group with a scorpion, that makes it a tough little mother. And so that's why I use the term badass, that we should be respecting what these worms have in common. And even us as worm researchers, we were underselling their wonderfulness. And so but I do it a little bit to sort of mess with the audience.
Yeah, well, it certainly is. It's a fun and provocative thing to say, and it reaches a lot of people, and I think that's a skill.
They might
Because
not believe it. They might think, well, this guy is too much in love with this weird organism.
Yeah. Yeah, I mean, they live in the dirt. I mean, there's really something to that, that they survive the winter, somehow. And but they're clear. That's one of the big advantages of this little worm is in the microscope, you can see all their cells. And so they're like a little piece of crystal, that's fluid and moves around. But they can survive from freezing to 25 degrees centigrade, and they develop perfectly regardless of all these conditions. You can starve them while they're developing, and then they can resume development without making any errors. Just a exceedingly robust little animal that has to live in the dirt.
And the field was founded by Sidney Brenner. He's not the first person to work on the worm, but he elevated the worm to be something that people should really care about for a lot of good reasons and intuitive reasons. One of them was his intuition about being able to do genetics on a fast time scale. A 3-day generation time is pretty close to what it takes to grow E. coli overnight. And he had been an E. coli geneticist. And Brenner was like one of the people who solved the genetic code. I mean, he's this iconic figure in the history of science. And he elevates this organism from something that was worked on at an agricultural field station to being an experimental organism because he's just a visionary. And we were followers.
Yeah.
We did, what were we? Postdocs number 50 and 80 or something in the field, But a lot of good people were like, hey, if Brenner says it's a good thing to work on, let's do it. And mostly we were following his lead. And boy, was he right.
Right.
And you were also following the lead of Bob Horvitz, right, from MIT.
Yeah.
And he, Victor, according to you in your Nobel lecture, he said that the worm is a tiny person in disguise.
Yeah.
What did he mean by that?
Well, he meant that the animal develops, from a single cell egg through embryogenesis, and then it develops as a larva, and that involves cell division, cell migrations, programmed cell death, differentiation of cells, control of stem cell lineages, when cells divide, when they differentiate, and so forth. And it's solving all the problems as it develops that any animal has to solve as it develops, including humans. And then once it becomes an animal that's mature, it behaves in ways using its brain and its nervous system that involve behaviors that it needs to survive. And it feeds and it browses and mates and communicates with other worms and so forth. It's using all the same molecular machinery, right, that humans use, but humans use it in a much more elaborate fashion. So if you trace the genome of the worm and compare it to the human genome, it turns out that most of the genes are the same. They're coding the same proteins and they're performing similar or sometimes identical molecular functions. It's but how they're regulated is what makes the worm different from the human. So that idea that the same genetic toolkit, as it were, is being used by humans and by the worm, because we had a common ancestor, and so we both inherited it from them. So that's what he means, that it's a little worm, it's a little human in disguise. It looks like a worm, but it's using a lot of the same machinery. And so, for example, Bob discovered the mechanisms of programmed cell death, or apoptosis, using the worm, and that's what he got the Nobel Prize for. He discovered it in the worm. So in other words, the machinery first emerged from the worm, from genetic studies in the worm, and then it was found to be the same machinery in humans. And again, there's the same kind of, the microRNA discovery of Let7, for example, once again, found first in C. elegans, and then Gary found it was conserved in humans. So again and again, that notion that it's a little person,
Who is the common ancestor between us and the worm?
Good question. I don't think we know from fossil evidence what it would look like, because it existed, right, Gary, before hard-bodied large animals. So it was probably a little animal, maybe no bigger than C. elegans, but it could do a lot of amazing things, some of which C. elegans cannot do. For example, probably our common ancestor of all the bilaterian animals could do whole body regeneration. But today, the worms doesn't really regenerate very well at all. But of course, we as humans, we regenerate, of course, very well, because we heal our wounds and regenerate tissues all the time. So C. elegans, I guess it's lost some of the attributes probably of the common ancestor, and probably gained some special of its own.
And I should mention for our listeners as well that this worm, C. elegans, elevated by, is it Brenner or Bremer? Brenner, by Brenner, and studied by less than 100 scientists closely, and the worm responsible for the win of these two scientists with me today is responsible for has been the background of a Nobel win 3 separate times. Has been kind of the scientific material for.
I think there's four. I think there's 4.
Oh, including you.
Is, but...
Okay, 3 before you guys and 4 with you guys, and one of them was Bob.
Yeah, it was Brenner, Horvitz, and Sulston, right? And then that was one, and then
Mello and Fire.
Mello and Fire was another one, and then Marty Chalfie,
yeah,
EFP, was a third one, and then this one.
Okay, thank you for that correction. So let's go back then.
Now there's about 3,000 people in the world who work on it. I don't know if that number is exactly right.
Okay.
But there's worm meetings, which you haven't experienced nerd unless you go to a worm meeting.
Oh, I have a question about the worm meetings. I can't wait to talk about the Boston area worm group and the Minnesota Worm Magazine, and that's coming up very soon. But so let's go back to the big discovery of the worm. Okay, so you two met in Bob Horvitz's lab at MIT. What year was that? Does it like a roundabout?
82, I would think.
82?
Yeah.
Okay. And you were both postdoc research fellows working on the worm. And by the way, I just want to say the reason I said 100 folks working on this is just based on what you said, Gary, about 50 to 60 postdocs or something. But that was an old figure. Obviously, now there's 3,000. So I just want to clarify for anybody who's wondering about that. All right. So you two met at MIT. You were both working in Bob's lab. And that's because Bob was working on it, right? So Bob was working on that. You guys came and you joined that party. Victor, you started investigating a worm gene called Lin4 under Bob's tutelage. That was this gene was partially responsible for egg laying problems in these worms, right? So does that mean that some of these worms were infertile and you were wondering why are some of the worms infertile and others are not?
Yeah, the egg laying defect is it's a little sneaky and hard to understand at first because they're not infertile because the animal is a self-fertilizing hermaphrodite, and it's the kind of hermaphrodite that fertilizes the gametes internally, so it makes sperm and oocytes internally, and they get fertilized internally. The
Interesting.
animal, yeah, so it doesn't need to mate.
Anybody else?
Yeah,
It
it doesn't
needs
need
itself.
anybody else. Now there are males, which conveniently, so we can do genetic crosses, but the standard way of propagating, let's say a mutant like Lin4, is just by self-fertilizing. And what happens is the animals, they can't lay eggs, so the eggs are fertilized and then they hatch internally, and then they consume the mother, called matricide, and then they crawl away, the little larvae. So that was a mutant that was isolated by a human being, looking through a microscope and spotting an animal that looked unusual, right? Because it was like a bag, a cuticle filled with larvae, and so somebody picked it, and that was back in the in the Brenner lab back in the 1970s that Lin4 mutant was picked up.
Wow, interesting. So the worms were not infertile. They were like they had babies growing and hatching inside them that would then eat the mother and just go and live their own lives.
Yeah.
Wow.
And by the way, just we had a dinner before Stockholm at the Swedish embassy in DC, and the MC at the dinner went on a whole riff about self-fertilizing hermaphrodites as a kind of pornographic all about sexual identity and about what is the worm doing with its self-fertilizing. Do you remember this, Victor?
Yeah.
He went on a whole riff about it that
Very
was
clever.
hilarious.
Yeah, it sounds like that's funny. It's daring, but it sounds very
daring.
funny.
It was really, but it was meant to be funny, and it made me laugh. I'd never gotten a humorous bit out of that before.
What's cool about the egg-laying phenotype is actually worth emphasizing how important that was, because Bob Horvitz did screens for egg-laying defective mutants, right?
Yes.
And Lin4 was only one of them. Also included in them were Lin14 gain of functions, that Gary worked on, but also dozens and dozens of other genes, including genes in the RAS pathway, the Notch pathway, right? Chromatin remodelers of all kinds, because you can end up with a worm that can't lay eggs by virtue of developmental problems, neurological problems, neural wiring issues, programmed cell death issues. If you kill the hermaphrodite-specific neuron in a hermaphrodite when it's only supposed to be killed in the male, right? That makes it egg-laying defective, and that was a very important. They found EGL1, which is an effector of programmed cell death. It's universally conserved. You see, so these egg-laying mutants, were amazing. They're a straightforward phenotype, bag of worms, right? But under that rubric, lots and lots of biology came out. At least egg laying, programmed cell death, microRNAs, RAS signaling, Notch signaling. So when we say the worm is badass, all this fundamental biology was uncovered by folks, identifying, straightforward, easy to spot mutations. Really cool.
Yeah, it's the worm that keeps on giving, and that explains why you two are still working on the worm now. We're talking about something that you started working on in '82, papers were published in '93, and here we are in 2025, and you guys are still working on the worm, and there's still more to.
Well, and it suddenly became much more efficient with genomics.
Yeah.
So worm genetics in 1982 was really pretty hard and boring in many ways, because getting the mutant is easy. And so that's the cleverness, is always to know what mutant to look for and to get it. And that happens pretty fast. But then getting to what the gene was a deadly year off your life. And in fact, the Worm Genome Project was started by John Sulston, who also won the Nobel Prize with Bob Horvitz. And I like to think that I was one of the reasons that he started it, because he looked at me when I was trying to walk to a gene, and he said, what a waste of a brain. That you should, all these people that were pretty good scientists were just spending a year to get to the gene they cared about. And he said, I'm going to parallelize this and do it on all genes at once. And he started working with just he and his technician, Alan Coulson, and they sort of put together a prototype of how to do the worm genome and then presented that to Jim Watson, the famous Watson of Watson and Crick. And at the meeting we were at in 1983 at Cold Spring Harbor, I think it was, and they had an audience with Jim Watson, and he goes, "Yep, let's do the worm genome." And they became super efficient. So now worm genetics is about 100 times easier than it was.
Well, with CRISPR genome editing as well, it's a whole yet another
Yeah.
stratum of amazing development. I mean, it's unbelievable. You like with so many organisms, C. elegans is, I think it's probably almost more efficient to edit a gene in C. elegans than in any other organism I know, because you can imagine an edit that you want and order the reagents, and that's the rate limiting step is waiting 6 or 8 days for the reagents to come, because then after that you inject the worms and a week later you have the mutant. So it's the change from 1980s, right, and then to the 2000s when the genome came on board, and now with the editing, like CRISPR gene editing, it's just amazing. It's almost indescribable. I can't really capture in words, how transformational these steps were.
Is there something else that would be great if it happened over the next 10 years that would take that even further?
That's a good question.
Like, is there anything else that the brain is being wasted on now?
Yeah, who knows? You never know until it happens.
Yeah.
Maybe other model organisms. I think that there's a promise now that that organisms of other clades, let's say, would be, be amenable now to rapid genetics, and genome editing and so forth. It would be great, and I think there's sort of people working on it. I have a postdoc in the lab, for example, working on the A-seal, which is a very distantly related invertebrate. And it can do whole body regeneration and so forth. And he's trying to develop the CRISPR tools so that you can really do the genetics on microRNAs and other, regulators in that system. And I know there are people working on octopuses and shrimp, and where the idea is to develop it into a model organism, where now you can, in the sense that you can actually do efficient experiments and ask questions about that organism's development.
Right, the same way that you can about C. elegans. Yeah,
right.
Yeah.
It's never going to be as efficient, obviously, some of these organisms would be, but the idea is to make them tractable. So now that you can do the kind of really, sort of deep exploration of mechanism that has been possible in C. elegans.
Mm-hmm. So getting back to Lin4 and Lin14. So Victor, you were essentially tasked with Lin4, and then a scientist named Chip Ferguson, I read.
Yeah.
You said that Chip Ferguson made a link between Lin4 and Lin14. I didn't exactly understand what that link was. Can you explain simply?
Chip, for some reason, he was growing Lin4 mutants, right? He was just growing them up, and and they grow on petri dishes and they eat bacteria. And so it actually, on a petri dish, it's only about a 60 centimeter or whatever across, 60 millimeters across. You can get thousands of worms to grow there. And so he just accidentally noticed that his culture of Lin4 was no longer egg-laying defective. And that's meaning that sometime in the last couple of weeks, let's say, when that culture was growing, there had been a revertant arose that had a mutation that suppressed the Lin4 phenotype and made the animals essentially wild type. And they're crawling around laying eggs and making lots of progeny. And so they had overgrown his culture. And so that's the discovery Chip made, that accidentally a suppressor had arisen. And that mutation happens to be a loss of function of Lin14, right? And so Chip mapped it and identified this new gene called Lin14, defined by this new mutation, which actually compensates for the Lin4 mutation. And of course, we understand now the reason is because the Lin4 mutation causes Lin14 to go too high. And now if you mutate the Lin14 protein, it kind of balances out.
Got it. Okay, so in a way, this was the precursor to Gary's entry into the
Yeah,
lab. Yeah.
yeah.
So, and then Gary, you came to Bob's lab. You kind of took over Lin 14. What happened to Chip? I want to know.
Oh, Chip got his thesis. Yeah,
He
yeah.
was working
He
on
was
something
working
else.
on another story. I think Chip's story was more broad. It was about egg-laying defective mutants broadly, and he identified lots of genes and organized them and so forth. And Lin4 was just one that he was working on, and Lin14. So
Yeah.
his thesis was already mature by the time he actually discovered Lin14.
And just parenthetically, Chip used to give group meetings about his SynMUV mutations. MUV stands for multivulva, which you don't even want to go into how these are named. My wife thinks anybody who works on multivulva, there's something wrong with them. But he would give group meetings about these MUV mutations, and we now work on them. Completely unrelated to microRNAs. They have everything to do with tiny RNA pathways, and I find it just poetic that something I was hearing about in group meetings that confused me. It wasn't like I was, oh boy, I hope I get to work on that.
No, yeah,
But boom, we backed into it, and it's totally awesome. And by the way, Bob Horvitz published probably a dozen papers about these SynMUVs that were not his most famous work at all. It's like number 10 on his list.
Yeah.
And they're wonderful papers.
Yeah. Yeah. The amount of biology that's come out of Bob's lab is
it's astounding.
Astonishing.
Yeah. It's so good. And he's our teacher.
And your friend.
And he was at Stockholm with us.
Yeah.
Beautiful.
Which was fantastic. It was, and he couldn't have smiled broader than with Victor and I getting this prize. He was so elated by it.
Aw, that's so sweet. I'm so happy for the three of
you.
you.
We were his first postdocs, the two of us. Right?
Wow.
Yeah.
Yeah. And I don't think at that moment he was predicting Nobel Prizes for us.
No way.
Why do you say that? You guys weren't. Why do you say that?
Superstars. We were fine. We weren't terrible, but there was nothing about it that made it seem like, oh, these guys are walking on water.
Yeah. I mean, I think that's an important aspect of this prize, is the fact that this is not a prize for scientific visionaries, right? This is not a prize for people who saw a problem, right, and devised a way to go get it, right? This is a prize for people who, Gary and I were just trying to do the best we could to do, the molecular biology of the day, to figure out what the products of these genes were, where when you mutate them, you get these developmental timing phenotypes. It's just that, the mutants are interesting. The genes must be interesting. They're probably regulators, but there was no theoretical reason to imagine that they should be anything different from what anybody knew about, you see? So we were just lucky, period.
We were part of a renaissance of developmental genetics, and that's how we thought of ourselves. And our role models at that moment were fruit fly geneticists, I would say.
Yep.
And we would go to conferences and hear really wonderful science from really fantastic people who work on fruit flies, which was the sort of model genetic system for 50 years, 100 years. Yeah. And that's who we were imagining. I was imagining we were going along that track, and then we sort of got into a different track because that's what the genetics told us.
Well, the developmental timing mutants were unique at the time anyway. There were no heterochromic mutants of fruit flies, right? So I think that was...
But we had a certain amount of shame about that too, because there was like, what's so mysterious about timing compared to how do you form spatial domains? How does a different segments of a fruit fly look different from each other? That seemed like a huge mystery.
Yeah, there was a certain amount of, let's say, trepidation, but some sort of concern about whether, or let's say concern about how relevant the worm would prove to be, right, to the bigger picture of trying to understand how genes regulate development, in animals. And because the development is so simple compared to like just a fruit fly. A fruit fly has hundreds of thousands of cells, and the C. elegans basically only has 1,000 or thereabouts. And the cell lineage was worked out by John Sulston and Bob Horvitz, meaning that they could trace the cell divisions of every cell in the animal from the single cell to the 550 cell larva, and then to the 959 cell adult or whatever it is. And since it was so simple and so reproducible, right, it felt like it was scripted in a way perhaps that was different in some fundamental way, let's say, than the way an insect or a human's development would be scripted.
Like it was too easy almost,
Yeah,
or
yeah,
too
maybe it's
small.
using this. It's not using a lot of the like as Gary alluded to, sort of the spatial information systems that flies were using and vertebrates were using. You worked on Hox genes, Gary. I mean, there was a question, right, to what extent Hox genes would be doing analogous things than what had been shown to be the conserved roles in flies and humans. Yeah.
Was so then in the community then was Bob and Brenner, were they kind of like, oh, those are the worm guys working on the worms, they got their postdocs, nothing's going to come from that? Was it a little bit like that? Because I
Well,
know
Bob,
obviously when the 1993 paper came out, it wasn't a huge deal in the science community, but it was a big deal in the worm community. Obviously Let7
that was big.
was different, but I mean, like Gary mentioned the word shame, you mentioned the word trepidation. Were you guys kind of like the underdogs at that
Yeah,
time in a
yeah,
way?
right.
We would be invited to meetings, so we weren't like ostracized or anything. We
No,
were
right.
part of it, but it was always a little bit of a distance because it was simple, and and there hadn't been any big hits out of the worm. And I think another thing that caused some trepidation is that there was a view that Brenner was distancing himself from C. elegans. So,
Oh,
Brenner was this
okay.
very quixotic character, and he was very special. And I barely knew him. It's not like he was a buddy of mine. I probably exchanged 20 sentences with him in my life.
Hmm.
But he was definitely the founder, and then he moved on to other things, and just because he wanted to solve the brain, and I think he decided that the worm brain wasn't that interesting. I don't know what he decided, but he moved on.
Hmm.
And so we had lost, but Horvitz was was our trumpet
Cheerleader.
player. He was always running around giving talks and
He's a believer.
proselytizing, and
Yeah.
and cell death was a big deal, but he also worked on RAS signaling, and RAS is an oncogene, and that was, people cared about that. That has something to do with cancer. And so he was very famous for that.
And Gary, when you came to the lab and you started working on Lin14, you were already versed in evolutionary conservation of genes. What is it? Because you had already done some work on that previously to arriving at MIT for the postdoc, but specifically, what were you trying to figure out about Lin14? So Victor was working on Lin4, and then they said, okay, you've got Lin14. What was your job at that point?
Well,
what were you trying to figure out?
They both had similar lineage changes, and one, it turned out, was sort of in the back end of the gene. It's called the 3' untranslated region, and that's sort of beyond the protein coding region. And that's what Victor and I figured out, that UTR was important. And then he figured out that Lin4, and so it made natural sense that Lin4 and this back end of Lin14 were talking to each other. And so that famous phone call that we made, we've made it famous by referring to it a lot, is sort of where we said, look, you can see the base pairing, you can see how they match up. And it just was sort of obvious from looking at the DNA sequence of these things that they're kissing each other. And Victor's lab figured out that it was this teeny tiny RNA. And that was, an amazing accomplishment, because who would think to look at 22 nucleotides long? Nobody would think to look that small. They did.
So what was that phone call like? Because you guys basically sent each other the sequences and said, all right, let's take a look. We'll talk to you in 20 minutes. Okay, I'll call you. I'll call you back. Okay, I'll call you back.
A couple hours. Because
hours.
Victor had a newborn at home, right? You probably had
really?
Matthew on your lap.
Yeah.
On the phone call.
And when was this?
It was right when you were at home with Oh,
it was 1992.
Early 90s, yeah, because the paper came out in 93.
I bet he
yeah, Greg was 2 years old. Right. Yeah. Right. Yeah. Yeah. yeah, I can't remember the chronology. We must have had a meeting during the day because I just remember it was a one to, person to person. We were talking, and you said something about we should compare sequences.
Yeah.
And then I think we said, oh, okay, let's send it to each other and then call later or something.
Yeah.
I can't remember
And
the
we have an email that we talked to each other, and that was a 1992 email,
yeah.
which is kind of early email days, by the way.
Yeah, you had to turn a crank.
It's close. Like, what's your first email, Victor?
I
Probably
remember.
sometime 85, 86, when we were postdocs. We
Yeah,
probably sent
probably.
one or two emails.
Yeah, I mean, the call was, I remember, just being, you know, having a sense that this is it. We figured, we know something here, basically. This must be how it works because Gary had shown that the sequences that was complementary to Lin4 were conserved in this other nematode. So the evolution really said that it's not accidental, and also there were multiple sites. That can't be accidental either. So there was this great sense that, hey, this is the way it works, antisense base pairing. That was really cool.
And so you guys were like, all right, this is the first
Yeah.
microRNA and its target
Yeah.
gene.
I also remember being really happy that we had actually done the phone call and that we had, found it together, sort of. And because, that's something if you share something, man, you can't take that away. It's just really, really, really special. And then there's also no concern about, I don't know how to say this, Gary, but about well, who found it? Who found it? Right? We found it, so we didn't have to worry about did Gary find it before me or did Victor find it before Gary or anything like that. We didn't scoop each other or
Yeah.
It was just perfect. If you're going to be two people in different labs where maybe you're competing, maybe you're not, if you bring stuff together and have that code discovery,
Yeah.
it just feels so enormously better than all the alternatives.
Yeah.
You know,
Because every journalist that has interviewed you guys since the Nobel win says, were you guys competing? Was there competition between you guys? And the truth is there wasn't, right?
No.
You guys were. And so why did you decide to take these respective pieces to your own labs after leaving Horvitz? Lin4 and Lin14. Like, why did you continue working on this? Like, for example, I said, what happened to Chip? You said Chip moved on. He worked on something else. I'm guessing scientists move on all the time, right? Why did you stick with this?
Well, I mean, one thing, Bob said we could and we should, right? I mean right? You guys should launch your labs working on it.
Oh, the labs were based on this work.
Yeah.
Got it. Okay.
I mean,
Bob, there is a type of postdoc advisor who doesn't let things out of their lab.
Yeah.
That's
a different
rare these days.
Different universe would have said, I'm keeping this stuff here. You all got to figure out what to do in your own labs. You can work on it, but I'm going to keep working on it, right? So we'll be writing grants to compete and all that jazz. So Bob was letting people go. I mean, we were his first postdocs. He could have done this quite differently. So clearly, he adopted a mode where I'm going to catalyze people's careers and then try not to step on them. So that's the first thing. And Gary, maybe it was just a pragmatic thing, that it really took so much work to clone a gene that it didn't make sense for either of us to try to do both of them.
Right.
Victor, you've often credited your wife, Candy Lee, the first author on your Lin4 paper, as the one who did most of the work on Lin4 in Bob's lab as a research assistant. And I'm guessing, did she go to your lab as well?
Well, she was in my lab, actually. Yeah.
Your lab. Okay. So she wasn't in Bob's lab.
Yeah. So in Bob's lab, Gary and I did all the work ourselves, but yeah, then when
she continued in your lab.
Yeah. So a couple of years after I set up my lab, Candy or Rosalind Lee joined my lab as my lab manager. And as it turned out, it was really, it was hard to get people to work on Lin4 because it was kind of an oddball gene, meaning there was only one mutation out there that anybody knew about that knocked out this gene. So that's kind of peculiar, And so I did get a postdoc to start working on it, but she, changed her mind about what she wanted to do, and so she left the lab. And then Candy came in, and because she was a technician, there was really no, she, if this didn't work out, that'd be fine. It's not a grad student's thesis. It wasn't a postdoc's career at stake. So it was a kind of a higher risk project, at least from our perspective. So that was really great, and then she did beautiful work. And Rhonda Feinbaum came in about a year later, and then they're listed as equal authors on that paper.
Okay, so we want to mention Rhonda too.
There's parity, really.
Yeah. But you didn't marry Rhonda. You did marry Candy.
No, I was already married to Candy.
You were already married to Candy. Okay, so you hired your wife.
It was nepotism. That's right.
My question was, did this worm project bring you together? But in fact, it didn't.
Something
else brought you together.
That's the
Yes. Oh my gosh. Exactly. All
Still
right. So
together.
Still together. Yes. And we're in, many pictures together on October 6th or 7th when the prize was announced, hand in hand, more than a wife, but a first author, right? Well, first co-author with Rhonda. So hello to Rhonda if she's listening. All right, so back to the worm geeks that Gary was talking about at the worm meetings here. So most of the scientific community maybe wasn't so excited about these 1993 papers because this the gene product of Lin4, the microRNA, they thought it was just a worm thing. This is just a worm thing, and those worm guys are doing their worm things.
No, actually, the developmental biology community had that response, and that's a big community.
Okay.
But there was also an RNA community that was growing in strength and discovery, and they were totally elated by this stuff. So we would be invited by all kinds of people who worked on regulatory RNAs.
Yeah.
Oh, cool.
It's not about developmental biology. It's about what do RNAs do, and they love this stuff.
Yeah, because
that was fun to encounter because we weren't part of that world.
Yeah.
Very nice. So you had some fans there. So it was just the developmental biologies that missed the boat, and now they're
but we
kicking
learned
themselves
from those fans, right? So we became part of the RNA community.
Yeah.
And it's a super interesting, highly developed group of people.
Yeah.
Things like the ribosome, right? The ribosome uses ribosomal RNAs and tRNAs. So translation it's the most wonderful thing in all of biology. And we got to engage that community by accident.
Yeah. And did that community and the findings from that community, Gary, influence your work on LET7?
Yes. Because the most conserved molecule on Earth is the ribosomal RNA. And, whenever they show you the tree of life, right, you see it over every third paper has the tree of life. And here's the bacteria, and here's the archaea, and here's the eukaryotes. That tree is based on ribosomal RNA and how much it changes or how little it changes. And so, that's the clock by which, we know 4 billion years of history.
But when the papers came out earlier, you mentioned, the worm meeting in Boston for worm scientists. And then there was, I heard you guys say on a podcast, one of you said a Minnesota Worm magazine where researchers published their work. I think that you guys were like gods to these two groups, right? When these two papers came out, I mean, what was their response? No,
we were members of the community. I don't think we were just fine.
Yeah. The Boston area worm meeting is a meeting of scientists from the Boston area, who work on C. elegans. It's been going on for a very long time. But
the gods of that world were Bob Horvitz, Marty Chalfie. We were very low level gods. What would you call us?
I don't think
little pooty,
disciples.
Little angels.
Disciples.
We never even set foot on the the foothills of Mount Olympus.
Yes, that's we were in some valley.
Yeah, actually, you should know that people really didn't take Gary and me very seriously, but until recently, But then the Worm Breeders Gazette is what you're referring to, right?
I see. Okay.
Yeah, and that was pretty amazing because that was a what was it, every quarterly or every, Bob would come around and say, we got to send abstracts to the Worm Breeders Gazette. And so you had to type up whatever you've been doing, and you'd send it off, and then it would get mimeographed,
Yeah.
right? Yeah, it would, they would copy.
Before the internet.
Yeah, before the internet. And you'd mail them off, and then Don Riddle and Bob Herman were running these things out of Minnesota. Yeah, and they would bind them up, and then you'd get it back in the mail, the Worm Breeders Gazette. And it would have like 60 or 80 pages, and each one would have an abstract of what the latest what's going on in somebody's
But
life.
very different subjects. So they do one thing about how a muscle works.
Unpublished stuff. Unpublished,
Another thing on how gametes work, another thing on fat, all different kinds of subjects. And that was one of the great things about worm biology, is it's a lot of things at once.
Yeah, and the sharing of information. We became, conditioned to a way to approach science where you just sort of share information, and because of the community led by, Sulston and Horvitz and others, had created this ethos of sharing and sort of community resources and jazz like that. And Andy Fire, for example, he made all these vectors, and he just for free he'd send out these 96 well plates with all these different worm vectors, worm plasmids, and he just send them out, to labs. And so you got stuff would come in the mail for free, that was resources. And so you just started to realize, wow, this is a great way to do stuff. Great way to do science,
Together, yeah.
Yeah.
And is the Gazette and the worm meeting still happening?
Oh yeah.
The Gazette is not really, as you can imagine, with the internet, the Gazette kind of became obsolete, right?
Mm-hmm.
The worm meeting, of course, is every year. I mean, every 2 years, it's international worm meeting. The next one is going to be UC Davis. The last one was in Glasgow, Scotland. About 1,500 to 1,800 people come to that.
Very cool.
Yeah, and the scope and range of biology is across everything you can imagine, because any biological process folks are interested in, they kind of seem to have picked a way to do it in C. elegans.
And it's only C. elegans, right? There's no other worms that are being studied here.
Oh, there might be a little bit of parasiticness in that.
Agricultural worms.
A little bit.
Agricultural or medical interest.
Mm-hmm.
Some pathogenic worms.
Mm-hmm.
There's sessions on those. On that.
A lot of genome comparisons.
Yeah.
Okay.
So the community is very sophisticated about genomics.
And so after the 93 papers, the big paper after that was obviously Gary's papers in 2000, I believe. And that there were two papers published in 2000, one after the other, I think. First announcing the discovery of the second microRNA, LET7, and later the watershed paper that announced that LET7 was conserved across species, found in a wide range of animals and humans. Gary, before these papers were published, in the work that you did to get to these insights and get the papers published, you, by your own admission, charmed researchers around the world to send RNA belonging to sea urchins, fruit flies, and mice via FedEx to your lab, and you found LET7 in all of them. What were all of those individual unpacking FedEx moments, and doing the work in the lab and seeing that it's there? What was that like for you?
Yeah, well, I didn't do those experiments. Amy Pasquinelli did them, and she was in Stockholm, which was very great to see her. So I did the charming to ask people to send the samples, and it was, telling them why we wanted to look for LET7 in their sea urchins or their sea anemones or clams, and it was just fun to get the range of organisms, sponges. And so they would send RNA to us, as a pellet. You don't even have to freeze it because it's just a precipitate. And then Amy would load the gel and probe it with the same probe and see, and boom, you get the result in about a week. And she got all these positive results. And then we put that together in a paper. And I remember the one disappointment I had was that we had a collaborator from Australia who I imagined wearing a folded up Aussie hat with a deep Australian accent, and he was an American emigre to Australia, and this was deeply disappointing to
Oh.
me.
Yeah.
Victor, you were all, there were skeptics, right? That people that thought microRNA was only, scientists that thought microRNA was only a worm thing. It was not going to be found in humans, right? Gary found it in humans. The paper published. You didn't know about the paper. You read the paper along with the rest of the world, and actually at that moment and before, well, not at that moment, but before that, you were one of the skeptics, actually, interestingly, right? You thought that was also just a worm thing. I've read that you've said that. And you had to, after reading these findings, you had to sit down, take a moment with yourself, look out the window and kind of absorb the news. What were you thinking looking out that window?
Yeah. I mean, you say I was a skeptic, and I've said I was a skeptic. And the thing is, it's really hard to gauge how I felt before Gary's paper. I think we were optimistic,
Okay.
and because Candy reminded me that, she did all these blots with, she did similar things that Amy was doing, but with Lin4, right? And, looking for homology in other organisms. I think we just looked at yeast and vertebrates, and there was no hybridization, and it turns out because Lin4 for some reason is not well conserved. There is a Lin4 gene in vertebrates, including us. It's just that the nucleotides have varied enough that you can't pick it up by hybridization or by blasting or anything like that. So we did sort of half-hearted experiments along the lines that Gary and Amy were doing, but they were half-hearted. I think obviously we were optimistic, but not convinced that I think once we got a few negative results, we said, all right, yeah, it's only in nematodes, So yeah, definitely. I definitely had to rethink it, but instantly I knew that wow, this is really cool. I mean, there's got to be more of them, So that was just so
well. And then there were the 3 papers that Victor did, and Dave Bartel and Tom Tuschl, looking for microRNAs by looking for something small. Those were paper of the year in science. It was a big deal.
And that was
the next year.
Definitely stimulated by Gary's paper, because we felt, whoa, if Let7's been around for 500 million years, there's been plenty of opportunity to evolve other ones, besides Lin4 and Let7. So we sort of knew they had to be there. And so Candy and I worked for like a year and a half or whatever it was, thereabouts, or 2 years, cloning little RNAs and sequencing them and stuff. And other people were doing it at the same time. Yeah, and so I told the story about how we just happened to be lucky enough to publish our paper at the same time as the Bartel and Tuschl labs, in the same journal of science. It was like super lucky, and that was really exciting.
Absolutely. And Gary and Victor, this is a question about Let7. So it was addressed to Gary, but it's certainly a question for you both. It's about kind of nomenclature of these microRNAs, the way that they are named. You guys.
Oh no, you're going to bring up the
long code, the code.
Mistake in my career.
Yeah. So we've got, the reason we love you guys is because, we love your work. We funded you guys. You went and won the March of Dimes Prize and all that. But also, obviously, you completely, and wholeheartedly inspired some of the work that we're doing now. We've got folks in London doing research on mRNAs and birth outcomes.
Mm-hmm.
And we've got a March of Dimes Prematurity Research Center at Imperial College London. And we've got researchers there that recently put out a paper that found 2 microRNAs, and one of them is a Let7 microRNA, as being overexpressed in women who had babies who were born small for gestational age. Mm-hmm. And I thought you might find that interesting. Your work has directly affected the work we do. The name of that Let7 microRNA is, drumroll please, HAS hyphen LE, not hyphen, but dash LET dash 7D dash 5P.
Yeah.
So to me, I've got a lot of question marks. What does that mean? What does this code mean?
Yeah.
Damned if I know. Nomenclature is hard. But let 7 I understood.
Yeah.
Yeah, me too. 7D.
Yeah, and the
Yeah.
HSA is Homo
Oh,
sapiens.
HSA, sorry.
Yep.
Yeah.
Yeah, LEP7, you'll notice that all the other microRNAs are named MIR, M-I-R, for
Yes.
microRNA.
Yes.
I think Dave Bartel or Tushel came up with that idea. Let's call them microRNAs. And then and I think I weighed in and say that's perfect, and we'll call it MIR, M-I-R, because I knew that MIR meant peace in Russian or something, right?
Oh,
Is that right, by the
I
way?
had no idea.
Yes, yes, it is. Yeah.
Because the
Yeah.
the,
Who
the
knew?
forwarding laboratory was called MIR, right?
It does. Yes, it does mean that.
Yeah. So
It
anyway,
does.
I thought that would be cool. Anyway, so everybody else is called MIR, but LEP7 was given the honor of being LEP7 forever, right? So
And Lin4,
yeah, no, Lin4 is MIR 125. Yeah,
but not in worms.
yeah, but yeah, but but in other animals. So even in other animals like
I didn't
humans,
know that.
now there's
Oh, I should be
LEP7.
more grateful.
That's to recognize the fact that it was the first one that was found to be conserved.
So
Oh.
LET7 is the only one that's HSA?
No. It's the only one
Let.
that kept the LET name.
Oh, the LET. Got
Yeah.
it.
Or
Instead
the
of the MIR. Instead of the
Yeah,
MIR. Got
Which,
it.
and the let is not very descriptive.
Yeah, it was lethal, but
Yeah.
Oh, okay.
yeah. So
All right.
yeah,
Go ahead, Victor.
yeah. The nomenclature is
Not important, it sounds like.
species, and then it's the family, LET7 family, and then there's a number of genes. There's a number of LET7 genes. So I think it's a D,
Yeah.
whatever.
And it's about also where it is in the genome. Is that right? It's about
The
location.
number
No.
of
Okay.
genes. There's multiple LET7s in humans in different parts of the genome. And then 5P just means that it comes from the 5' side of the hairpin. So.
All right. Well, thank you for answering that for me. All right. So this is obviously a big question. I don't know if you can answer, but I hope you can. What do we know about why microRNA levels differ among people and cause disease in some and not others?
I don't know the answer to that.
It would really depend on the organ. Yeah, I can imagine a lot of questions, right? One question I would want to know is like, well, what organ are we talking about? And then like in that organ, well, what cell type is the microRNA different, in level? Yeah, and you'd want to know also how much it varies from person to person, in a population that doesn't display the disease. So that's one of the things about microRNAs that's been apparent, is that there's an enormous amount of complexity around microRNAs.
Yes.
They are expressed in patterns that are tissue specific. They can be also expressed or not expressed depending on whether the cell type is growing or not and dividing, right? So I think in a few cases, right, there's been genetic links to microRNAs, right? And then perhaps you can say something a little bit more substantial, because then you could actually try to do some experiments with, say, cell lines, for example, to try to model the physiology.
I'm not aware of, like, if you go to Nomad, which tries to document all the human mutations that are out there in populations, I'm not aware of whether they look at microRNAs and what they do.
There is one MIR96, which is congenital blindness. And that's actually a misexpression of the microRNA, I think.
Oh.
And there's a few others. And of course, there's the Argonaut syndrome, where the mutation is in the Argonaut. So that's causing, disarray, let's say, in the function of many microRNAs. There's also microRNAs that drive tumors. Like there's a cluster, MIR 17 to 92 cluster, and that is it's duplicated and overexpressed in lots of different sorts of tumors. And there's a lot of work has been done in trying to figure out exactly what are the targets of these microRNAs such that they are driving the tumors, and that turns out to be a very complex situation.
Because there are a lot of targets, right? That's the problem. One microRNA can target hundreds of different recipient genes.
A cluster of microRNAs that's overexpressed. So there's 5 or 6 of them.
Right.
Um, yeah, and then there's a Dicer syndrome, right? Which is mutations in Dicer, which come up in Wilms tumor, and it's because the processing of a whole population of microRNAs is affected. And it turns out in these kidney cells that can be tumorigenic, but not in other cells, right? So that your question is so apt, right? Because you can have the same genetic disruption of microRNAs in a different, you know, tissue, and it doesn't cause the same effect at all. So it's perhaps all about what are the targets in that context, you know, in those different cell types.
I want to just move on to have a very short conversation about our solar system, if you guys don't mind. Both of you at a young age were interested in astronomy. Gary, at 6 years old, you were building planets and orbiting them around a model sun. And Victor, at 17, you were featured in an astronomy magazine for an eyewitness account about the 1970 total solar eclipse. You said it was the first time you felt like a real scientist. How did your love for astronomy lead you both here to developmental biology and genetics?
Good question, Gary. Yeah.
Well, that was my introduction to science, I think.
Yeah.
You know, we're both born within a year of each other, and we're sort of 8 to 10 years old when the space program is on TV once a month with a launch. And I learned a lot about rockets and things like that by watching space launches, and so did a lot of kids. And you know, when I was we would go to Safeway. I'd go shopping with my mother, and at the checkout stand for $1, you could buy the Golden Book of Science, which I think I might have here. Yes.
Yeah. Golden Book of Science.
Here we go. Okay, so this is the world of science. Why is it backwards? Is it backwards for you?
No, it's not backwards.
We see it.
Yeah.
Oh, I must be backwards.
Yeah.
So, you know, so this is, see the deluxe Golden Book of Science. You can even see my little 8-year-old name in there. That's me saying it's my.
Yep.
Aww.
Right. And it has pictures of planets and things like that. These are scales. These are all factors of 10 in size. Sorry.
Yeah.
Where are we? And I have to show you. This is, I'm sure I'm wasting your time.
Yeah.
But hold on.
Not at all.
Here we go. So, nope, that's immunology. Where are we?
Yeah, well, you said the world of science. That's really the key. Yeah.
Okay, so this was at the Safeway checkout. That is, let me get his name. It doesn't say his name. It's the great fly geneticist.
T.H. Morgan?
No, Lewis.
From E.B. Lewis.
Caltech.
Or
So
Lewis.
he's like a big shot.
Yeah.
And this was all part of the, you know, making Americans educate their kids about science because Sputnik was launched in 1957, where the Russians were winning.
Yeah.
And so the way we beat the Russians is by training Victor and me. We're the difference. That's why we speak English in the world.
Yeah, I mean, you said the world of science. That, I mean, astronomy was a nice, easy way, so to speak, or convenient and accessible way to open up the world of science to a young person. The space program is incredibly inspiring, but I think I was intrigued by I don't know, I just somehow you know, the telescope and the sky captured my imagination, and you know, I learned you could build a telescope, and that was just astonishing that you could actually make a telescope in your basement. And so it made you feel like you were engaged with science. You go outside and you could chart the stars and maybe even take a picture of a comet, you know, and make the telescope. I felt like a scientist. Right?
By the way, did you notice Venus in the last couple of weeks, just under the moon? Oh, sorry. Yeah, no, Venus has been incredibly bright.
Yeah,
I think, yes. I haven't known what it was, but I'm like, what planet is that?
That
It's
is
very
Venus.
bright.
It's
Yes.
super
right,
bright.
the sky is beautiful, you know. So Yeah, and then when I went to that eclipse, yeah, I wasn't featured in that magazine. I was just mentioned in a paragraph, but yeah, it was really cool because I think that's one of the things that is so important for youngsters. Is somehow to feel like they already belong to, let's say, this tradition that they want to join.
Right,
You know,
right.
it's not like that's a goal you have to somehow reach. And nowadays, there's
Mm,
actually so many opportunities for kids to do science at home. All the data sets from the Webb telescope, from the Hubble, these are all public. All the genomes are public. They're online. So if a youngster wants to do real science investigation, all they need is some sort of mentor, some sort of guidance to get in a framework of how to ask questions and jazz like that. There's tremendous amounts of data that are available. It's actually wonderful.
I got astronomy merit badge when I was a Boy Scout.
Mm.
Gary, you're still interested in space. You're looking for life outside Earth. Can you explain why you're trying to convince NASA to look at ribosomes found on Mars?
So the bottom line is that I told you earlier
Yes.
that the ribosomal RNA is the most conserved thing on Earth, and it's used as the molecular clock for classifying everything on Earth. And it's that tree of life that we know about starts within a couple 100 million years of Earth cooling down when it first was born.
Yeah.
And so most people think, well, that's how long it takes to go from a primordial soup of chemicals to evolving RNA and then DNA. And I personally think that's kind of dopey, and that the life came to Earth. And so as soon as Earth cooled down, bacteria rained down on Earth. And but I have, that's my own sort of bias. And if you had a room full of people who think about the evolution of life, 99 out of 100 would say that's stupid.
Well, I don't know. I don't know if they'd say it's stupid. I think it's a very, very good question, right? Is how long does it take to
Yeah,
evolve a ribosome?
yeah.
This is yeah, you talk to origin of life people, they have a lot of fascinating insights into sort of how, let's say, how rapidly things can happen, you know, um, and how much time there has been. But Gary's point about 200 or 300 million years after the Earth cooled, I mean, we don't have a theoretical way to really say, yeah, life could have evolved in that amount of time. And so, given the fact we can't say that, Gary's hypothesis is totally valid and completely up there. The problem with it is my problem with that hypothesis is that life came from Mars. Well, then how did it get to Mars? And I think Gary says, well, maybe it came elsewhere. Even if the universe is what, 13 or 14 billion years old.
It only gives you a
Yeah, it
factor of
only gives
three.
you an extra 10 billion years,
I
right?
think
You
the
know,
physicists are going to figure out that multiplied by they got their
yeah.
multiplication wrong, and the universe is 1,000 times as old.
So what you've touched on here is, I think, one of the deepest questions of biology, probably one that we're going to be working on indefinitely for centuries, right? Is how does life come about? You know,
What I'm trying to get is access to a Martian return sample.
yeah,
So building an instrument to send to Mars is hard to do. It's hard to get approval. It's a 30-year process to build something like that. I don't have 30 more years. But a Martian return could happen in a decade. It's probably not going to, but it could. And I just want to be one of the people that gets a little bit to
what
do.
about a comet return? You don't know.
Yeah, it's
You want something that came from Mars. Period.
So what would you do?
The,
Like take a
the further
shovel
the
and just
better.
take some dirt off of Mars or
Yeah,
some
yeah.
of the sand or something like this?
Well,
Well, they
they've already put into little tubes. I think they have 30 tubes of soil samples that geologists say
yeah,
this is a sample that was in a dried up lake, and
Okay.
that's where
yeah.
life would be.
The probe that's up there, the mechanical, they have the robot has gathered samples and cached them, right? And so the idea is that in the future we'll send a craft to land, pick up the canister, and bring it back.
But it's really hard to take off from Mars.
Yeah.
It's much different than landing. Taking off means it has to have a propulsive system that gets sent all the way to Mars and then gets back here. It's not trivial. And they
Right.
just had a whole rethinking of it, and then the cost structure is like 3 times more than they want it to be.
Mm. Important though.
Yeah. So I keep telling the people who SETI, right? They want to search for extraterrestrial intelligence, and they say, "Oh, I want to see them send pi. I want to see a radio signal that has 3.14159." And I say, "No, you should be looking for GATC. That's the ribosomal sequence. That's what you should be looking for."
Yeah. Yeah. That would
do,
be far out. Yeah.
do each of you have, that would be far out. I
Yeah.
love that. Do each of you have time for 2 more questions, or should we go into the last question?
No, I have time for two
Go ahead.
All right. So this is about more your most recent work. So Gary, back to you. You're also trying to figure out why humans and vertebrates, you touched on this a little bit also earlier. You're also trying to figure out why humans and vertebrates don't have some of the proteins that present microRNAs to their target cells the way some organisms like tick spiders and worms do. You say tick spiders and worms are very good at antiviral defense, and it may be because of those proteins, but humans evolved away from them. Have you figured out why?
Yeah, I mean the best guess, and it's just a guess, is that having a nervous system that thinks and can think about pi is not compatible with that kind of antiviral system. COVID makes you realize that we're not very good at antiviral compared to our worms are really good at. And remember that the antiviral pathway called RNA interference was discovered pretty much at the same time in plants and particular fungi and worms, all of which have this 2-stage RNAi system that uses what's called the RNA-dependent RNA polymerase. And that's what makes these organisms really good at antiviral defense.
The two
Really
stages.
good. Really good at it.
And that's the 2 stage, and we only have one.
Yeah. And so it's the badass organisms. That's the enzyme system that I'm calling the signature of badassedness.
They do some things better than us.
They do, right? Clams do it, plants do it, scorpions, ticks.
Why would the nervous system preclude us from having that?
I don't know the answer to that, but clearly the sort of animals that include us have really they've bet heavily on microRNAs as opposed to RNA interference. And maybe they need that for all the subtleties of being able to do calculus.
And Victor, your continued work on C. elegans has shed light on Argonaut syndrome. This is a rare neurodevelopmental disorder that's caused by mutations in proteins that help microRNAs silence genes, so that help them do their main work. Once these important proteins called agoproteins are mutated and incapacitated, they could cause disruption of microRNA silencing, turning on certain genes that lead to these syndromes. You recently found that these protein mutations can be as small as a single amino acid change. How can clinical scientists use this insight to help humans avoid these syndromes?
Yeah, I mean, of course, that was not our insight. This is an interesting situation where we were attracted to a field that had already been established as a clinical genetics field, right? It's fairly recent discovery of this Argonaut syndrome in kids, maybe 6 years ago or so. And what was peculiar about the mutations is, and the condition is discovered when a child displays developmental delay, and if the family happens to be in a medical system where they will do genome sequencing, then they can find out that in fact what the child carries is a novel mutation that the parents don't carry. So it's a new variant, and so one of the genes for one of the Argonauts is mutated, and it's always only one amino acid. That's what was so peculiar. That all the patients when they were sequenced, it's only one amino acid, but a different amino acid in a different patient. Although there would be some patients would have the same amino acid mutated, but so the Argonaut Alliance, got together, and it's Alliance of Families and the clinicians involved in these research projects got together, and they sort of published some papers describing these human mutations. And that's when we got intrigued, because we were curious about why is it that there always just single amino acid change causing this condition? Why isn't it a deletion of the whole gene, right? Um, and so we thought we would explore this in C. elegans by making that same mutation in the C. elegans gene for the same argonaut, the microRNA argonaut. And what's so striking about these proteins is that they're almost completely conserved in amino acid sequence between humans and nematodes. So they probably work almost entirely in the same way. And so the idea is if we put the mutation into the C. elegans argonaut and then explored what happens in the worm, we might learn something about sort of the nature of the molecular disruptions that might be going on in the humans. But importantly, at the same time, we would learn something about the function of this argonaut protein. The idea being that these mutations that come from the humans would be instructive about argonaut function, because they're disrupting the function in some fascinating way. And we figure that they're going to be instructive, and basically allow us to learn more about this protein that we are passionate about for other reasons besides the clinical relevance. And of course, at the same time, we have some hope that perhaps what we learn might be helpful, in a clinical context. And one of the things that's apparent is that if you were to actually remove the mutant argonaut, it would probably be better for the patient, because if you remove the mutant argonaut from C. elegans, the worms get better. They actually become wild type again. So I think that's the most probably relevant clinical perspective we could bring, is this confirmation that you would want to pharmacologically or somehow knock down the mutant argonaut, and that could help the patient.
Yeah, so that was going to be my next question. How could you do that in a human? You said pharmacologically, so they would take a drug, or would CRISPR be involved in any way there?
It could be probably a more direct way would be using RNAi, actually, because there's a number of RNAi therapeutics even in the clinic. So Alnylam and other companies have developed these therapeutics, which is the basic approach is to knock down your protein, a toxic protein, basically, by using RNAi. And these therapeutics have been developed even for delivery to the nervous system. So it's not science fiction, really. It's one could be optimistic that this could be possible within 5 to 10 years or so, if attention were devoted to it.
Fantastic. Well, thank you for your work on that. I'm sure the Alliance is very, very happy for those insights. Gare, this is the last question for each of you. Thank you for sticking with me for this nearly hour and a half interview. Gary, last question for you. In 1974, after your bachelor's degree, you lived in your van for a year in Oregon planting trees. And after that, you spent a year in South America. You've said multiple times in multiple interviews that trip gave you countless stories with which to regale the scientists in your lab at MGH. And I've been wondering, what's your best story? Have you got a good story you can tell in 2 minutes?
Well, there's a story of...
It's got to involve you.
Say it again?
It has to involve you. It can't just be like a story that you heard or something.
No, yeah. Well, so there's a story of in Bolivia, there was 3 of us traveling to a fiesta outside of Cochabamba, Bolivia. And this is on a plane, and it's about a 3-hour bus ride. And so we all go to this fiesta, and then we're on the bus. It's just a public bus going back, and I'm in seat number 1 and 2 with my friend and 3, and I noticed that we're going down a straight highway. It's not mountainous at all. It's a valley, going down a straight highway, and the bicyclists on the highway are like going off the highway because they see that there's something wrong with how the bus is being driven. And then I noticed, because I'm in seat number 1, that the eyes of the bus driver are kind of closing because he's falling asleep at the wheel. and
Yeah.
it's an intercity bus. And so I start yelling esquina, esquina, which means next corner, which is what you use on a city bus to get off at the next corner in Latin America. And
Ah.
he's like yelling at me, it's no esquina, this is interciudad, intercity. And I said, tu eres borracho, you're drunk, you're drunk, you're drunk. And he pulls over and he starts attacking, sort of punching at me. And in the story that I told and retold 100 times, I kicked him and knocked him out. And everybody on the bus is laughing their heads off at these gringos who just knocked out their bus driver. They think it's the funniest thing they've ever seen. And then I reread some letters 40 years later that I'd written to my brother, and it wasn't me who knocked out the bus driver. It was the third guy who was traveling with me who kicked the guy. But I had mutated the story. So it says a lot about me that I became the hero of the story. But it is a true story that one of the three of us knocked out the bus driver. And there was a marching band on the bus. And again, I can't make this stuff up because it was a fiesta. And the head of the marching band in his uniform drove the bus back to town. And then the bus driver was arrested for being drunk, which was not good. So I was kind of a hero, but I really wasn't the hero, except for saying esquina, esquina, esquina.
Wow. So the guy in seat number 3 saved the lives of everybody on that bus, including you, and allowed you to get the honor that you got in October.
Yeah.
Exactly.
But you also, I love another story you told once, Gary, is you were invited, there was a whale watching trip or something, friends were, and they said, we need a science officer.
They needed a biologist.
Oh, a biologist. Yeah. And you envisioned yourself as science officers like Spock. You would be
well. But I kept quoting from Moby Dick, and everybody on the bus was a kiss the whale type. And on a whale voyage, it's really boring, right? Because you don't see anything for 5 days, and then you see a sprout and a spout.
You were the biologist.
I was not much of a biologist for the trip, but it was free.
Yeah.
Victor, your last question has to do with your father. You've often spoken of your dad, how much he's inspired you, how much he encouraged you, and how you always looked up to him. He wasn't around to see you win the prize, but if he were, what do you think he would say?
I don't know. Yeah. I mean, that's a really good question. I mean, I think he saw Gary and me win some awards, and what struck me about his reaction to that was that he made it clear that he knew that wasn't what it was all about, right? In other words, he knew that I really was a scientist to be a scientist, and I was never looking for awards, and I was never looking to be famous and everything, and he really knew that. And so one of the great gifts I would say that my dad gave to me before he passed, in 2014, was to make me understand that he really he understood me, that he understood that I wasn't trying to get famous, and so that was wonderful. So I think that he would have taken it in stride, sort of, but of course he would probably marvel at the arc of his life and my life together, right? Because where he came from and what he went through during World War II, he was a slave laborer, and then he built a farm, and then a woodworking shop and a business, in Vermont, and raised a family, and then I have this improbable life, as a scientist. I think he'd be really amazed, and just marvel at what can happen, in the course of 2 lifetimes.
But you knew how to think with your hands.
Yeah, I mean, of course, he taught me, to be a do-it-yourselfer. I mean, I think he probably would have been proud, and probably he would have been able to point to things that maybe he kind of taught me. I mean, the idea that you can, just to do stuff with your own hands, try things that you are not familiar with. Maybe we, I don't know. It's a very good question, and I love to think about it. And then thank you for asking it, because it's very special. You know, all of us, and you know, Gary's family also has incredible stories about, and my wife Candy's story. She's an immigrant also. The stories of, and you, the people that whose shoulders we stand on, you know, and they're just sort of everyday heroes, right? Who live their life so often against incredible odds, you know, and you know, it's just staggering.
Survivor bias.
Yeah. Mm-hmm.
Yeah, and they all deserve a Nobel. But I want to congratulate you again on your Nobel win, Victor. Gary. Thank you so much for your time today. This has been really special.
Yeah, my pleasure.
All right, that was fun. Thank you for the interview.
Yeah.
You're welcome.